Skinny love just last the year.
Detecting anorexia nerviosa, Aptamer-based biosensor, the next step in preventative treatment. According to the Mayo Clinic, anorexia nerviosa is defined as an eating disorder primarily distinguished by abnormal eating habits, distorted perception of self-body image, and extreme fear of weight gain. Anorexia nerviosa has one of the highest mortality rates for any psychological disease and can affect any person regardless of gender, age, or economic status. In the past, anorexia has been categorized as a mental disease, chiefly brought on by myriad environmental factors such as diet and body shaming. However, evidence has increasingly shown a linkage between genetics and the development of the disease. An analysis of a genome-wide linkage of anorexia nerviosa was performed on family members of affected individuals, resulting in evidence that suggested marker D1S3721 on chromosome 1p to be a susceptibility gene for the disease. So how our aptamer will be different from previous ways of detecting anorexia through genetics. So genetic analysis has in the past only been used on relatives of individuals that are already suffering from anorexia nerviosa, and our biosensor would have the ability to detect genetic susceptibility before the development of the disease, and therefore we could enable preventative treatments. And in the past, it's only been genetic comparisons. There hasn't been any biosensors or aptamers involved in the perhaps preventative treatments of anorexia. So we would promote testing of individuals that don't have family member linkage to the disease.
So we decided to use single-stranded DNA as the form for our aptamer because, as opposed to single-stranded RNA or a peptide, single-stranded DNA should theoretically bond to the target anorexia gene on chromosome 1. Through the complementary base pairing, this should produce an aptamer shape change, fluorescence, and that should bring about an awareness to the reader. And we decided to choose fluorescence because fluorescence and the fluorescence machines are very, very highly sensitive. And what will happen with the fluorescence is you'll attach fluorescent molecules to the ends of the aptamer, and upon binding to the target anorexia gene, the reporter will fluoresce a light, and a signal transducer, a fluorescence transducing machine, will alert the reader to the target gene.
The biosensor that my group has developed will bind specifically to chromosome 1. This target was chosen because it has been shown that there's high non-parametric linkages on marker D1S3721. That there is a susceptibility gene to developing anorexia nervosa. So creating this biosensor will help those who are predisposed to this gene, will help allow them to get counseling and help before the gene takes effect and causes them to develop a severe case of anorexia. So in order to obtain the right aptamer for the biosensor to bind to, we're going to run a multitude of trials that gets each time, make sure that it binds to chromosome 1. So to do this, we need to make sure that it will bind to chromosome 1, and hopefully to marker D1S3721. So in the select process, we have to make sure that the aptamer will not bind to other chromosomes, DNA, and other components in blood samples such as hemoglobin, platelets, red blood cells, and white blood cells, because this could create a false positive, and we don't want that. So we can show that this biosensor works by performing an experiment on a group of participants who have been diagnosed with anorexia and see if they do have this predisposed gene, and test members of their family to see if they also have this gene, see if it's passed down genetically, see how exactly the gene works. So if this gene does work, fluorescent will signal will be produced from when it gets attached to the gene. So hopefully this experiment will create results that are consistent and proves that it actually works. But having the biosensor available to the public may potentially have downsides because they may take it and be shown that they do have this predisposed gene and begin to obsess over the fact that they may develop the disorder and develop unhealthy habits that could be harmful to them. But altogether, knowing that you may have this predisposed gene may help you in the long run to get the help and counseling or whatever you need to prevent getting anorexia nervosa.
The biotin and streptobiotin are used to obstruct the aptamer to the glass slide and immobilize it. It would be easier to read from the glass slide, and the biotin would significantly increase the stability of the aptamer. The aptamer will have a quencher and a reporter, and when it bonds to the target DNA, the reporter will fluoresce and transmit a signal to the doctor or the reader. The only technical hurdles we may face in creating this would be finding a proper fluorescing reading machine and having the aptamer bind to the correct part of chromosome 1. For our project, Molly did most of the writing for the project, including why locating the gene was different from proverbial ways and why it was more important to focus on anorexia. Haley and Vicky did a lot of the research for the project and provided us with most of the information in making the video. And I did the drawing for the aptamer, the makeup for Vicky, and the video editing.